Good morning. I'm here to welcome you to the third annual Biohealth Communications Competition. My name is Michael Furr. I'm the Vice President of Business Development with Winland Electronics. At Winland, we manufacture products and cloud solutions for monitoring critical areas, whether it be facilities, processing, or inventory particularly. We're happy to be supporting the Biohealth Summit. It's our first time, looking forward to many years to come. And as a member, we support Wisconsin's vast resource of growing the industry, especially through our students, which are you here today. And I'd like to introduce the Michael Harrison, who's going to moderate this situation. Thank you.
Thanks so much, Michael, and thanks again to Winland Electronics for sponsoring this session. Hi, everybody. Good morning. I'm Mike Harrison from BioForward. I'm the Director of Strategic Initiatives and Community Outreach. Title's maybe a little bit too long if I have to take a breath in the middle of it. I'm so happy to welcome you to this competition. So this is the third year we're doing it, and we've expanded this competition this year to include undergraduates. I can't tell you how excited I am to allow this kind of interaction with students and the industry to continue to grow and expand. Anybody that knows me knows that I really love to talk about science. That's why I was so interested in getting something like this going. My kids know better than anybody that if they ask me a question like, why do I get dizzy when I spin around? All of a sudden I light up and I'm like, this ride to school just got interesting. Buckle up. I know I only have a couple more years before they completely resent that kind of activity, but in the meantime, I love it. I love sharing ideas, helping people understand the world around them, and I really hope that the students that are in this competition feel the same way. I consider this to be an important talent, an important skill for scientists to secure the future of our industry. It's really hard for us to help those people that don't quite understand what it is that we do, why we do it, and as a result become, fearful or distrustful. I recently saw an interview with Carl Sagan, and in it he said, we have arranged a society based on science and technology in which nobody understands anything about science and technology. And he went on to say that that's kind of a combustible mixture of power and ignorance that could end up blowing up in our faces. And I truly believe that we have to explain what it is that we're doing to people so that they know that this is of benefit to society, so that they understand why the pills that they're taking help them, why the technology that you're working on in some lab that they've never been to, why that's going to change the world. So I encourage all young scientists to talk to anyone and everyone, because as soon as we're willing to share our passions with others and explain why this is so important to us, then we'll be able to build that trust and truly change lives. So we have two groups of students here that are going to be giving 3-minute presentations describing their work or scientific topics to a general audience. The first group is going to be our undergraduates. So this is the first year that we're doing this. And some undergraduates do have research projects that they can discuss. Others may not, or they may choose to talk about something else. We've given them the option of talking about any topic that they want that falls under the realm of personalized medicine, the theme of this year's summit. So I would like to just get things started here, and we'll keep moving along. The first person that is going to speak today in our undergraduate group. And before I get going, I will say that we had several videos sent in for all of these groups, and these are the top 5. So these are already winners. They've already won something. Everyone that you see here today is a finalist. So I hope you enjoy their presentations. First up, we have from the College of Agriculture and Life Sciences at the University of Wisconsin-Madison, Mia Kroll.
Is it this thing?
Yep.
Okay.
Just hit the green button to go forward and the red to go back.
Perfect. Okay. Hello, everybody. My name is Mia Kroll, and I'm a senior here at the University of Wisconsin-Madison. I am majoring in biology and obtaining a certificate in global health. And today I would like to talk to you about how personalized medicine will be used in the future to prevent disease. Thomas Edison once said, "The doctor of the future will give no medication, but interest his patients in the care of the human frame, diet, and in the cause and prevention of disease." So let's kind of dive into this. What does this mean? To me, this quote talks about preventative medicine. Preventative medicine is when a doctor works with a patient to create a healthy lifestyle to prevent disease and illness in the first place. So, how do we do this? Can we make a set of rules that every person has to follow in order to be considered healthy? This may be one idea, but will it work? Let's go over an example. The number one nutrient deficiency in the world is iron deficiency. So, do we start giving everybody iron supplements to take every day? No, because some people have hemochromatosis, a genetic disorder where they create too much iron in their blood. This is where personalized medicine comes into play. Personalized medicine involves an individual's genetics, family history, personal goals, religion, and environment. And they take it all into consideration to develop and tailor a personalized lifestyle plan for these patients involving nutrition, exercise, hydration, and sleep. How do we accomplish this? In order to accomplish personalized medicine, we need a variety of resources working together, including medical professionals, all sorts of technology, pharmacology, and even AI. Taking these resources and putting them together is going to help us make these amazing personalized plans for every individual person. Now we get to our main question. How does personalized medicine prevent disease? The future of medicine is not reacting to a disease, but preventing it in the first place. And one way we can do this is by certain genomic tests. A subset of these tests have predictive capabilities to them, meaning that they can predict a certain disease or illness you may get before you even show symptoms for them. For example, we may find a gene in your genome that is linked to ovarian cancer or breast cancer. And so then, as a physician, we can recommend an increased frequency of mammography, prophylactic surgeries, and chemo prevention therapies. Another example works with heart disease, the number one cause of death in the United States. We can prevent heart disease by lowering blood pressure, cholesterol, exercising regularly, modifying your diet, and keeping a healthy weight. And all of these factors work hand in hand with personalized medicine and your lifestyle plan. In 5 years, 73% of cardiologists indicate that personalized medicine will have some measurable impact on patient treatment. And in 10 years, more than 9 out of 10 cardiologists will believe that personalized medicine will have a larger role in cardiovascular therapy altogether. Benjamin Franklin said that an ounce of prevention is worth a pound of care. It's time to turn our focus on preventing disease. By the use of personalized medication, we can accomplish this feat as it is entirely within our capabilities. Thank you.
Okay, our next undergraduate is from the Department of Biomedical Engineering at the University of Wisconsin-Madison, Zach Spears.
Hello, everyone. Like you just said, I'm a senior studying biomedical engineering here at UW-Madison, and today I'm going to talk about the promise of personalized medicine in transforming lung cancer treatment. Lung cancer is the leading cause of cancer death worldwide, with 1.8 million people dying every single year. Despite this, 5-year survival rates remain incredibly low. Personalized medicine has the potential to offer better outcomes by tailoring treatments to each person's individual genetic profile. So what is personalized medicine? Personalized medicine is an approach to healthcare that takes into account each patient's unique genetic profile along with other unique characteristics to develop tailored treatment methods. This approach is revolutionizing cancer treatment by allowing physicians to pick treatments that will have better outcomes for their patients. We aim to increase effectiveness while decreasing side effects. Historically, lung cancer treatment has followed a one-size-fits-all kind of approach. We've relied very heavily on chemotherapy and radiation therapy. And while these are both incredibly valuable in a physician's toolbox, they can have limited efficacy at times and can have very severe side effects. So, the promise of personalized medicine is that we can tailor treatments to each patient's genetic factors that are driving their disease. We can use genetic testing to identify these mutations and then create those precision therapies. One example of a therapy that's on the market today is crizotinib, which was FDA approved in 2011 and is used to treat cancer patients with an altered ALK gene. There are several different types of precision therapies out there that are available. There are TKIs, there are immunotherapies, and there are PARP inhibitors. TKIs target specific proteins that are overreactive in cancer cells, whereas immunotherapies allow the body's own immune system to fight back. PARP inhibitors, on the other hand, don't allow the cancer cells to repair their own DNA, in turn causing cell death. Despite its potential, personalized medicine still has several challenges. The cost of the associated therapies, as we heard about from some of our keynote speakers earlier, ranging up to $2 million, and the associated cost with genetic testing are all factors we have to take into account. Additionally, the availability of these drugs and the complexity of analyzing the vast trove of genetic data has to be taken into account. But despite this, the future of personalized medicine is still incredibly bright, with advances in genetic testing and data analysis making it easier to identify those actual mutations. The future of personalized medicine is that personalized medicine can be the standard of care for lung cancer treatment down the road. We can have increased effectiveness and decreased side effects. To conclude, personalized medicine is revolutionizing lung cancer care by tailoring these treatments to each patient's unique genetic profile. Precision therapies offer hope for better outcomes with fewer side effects. And while there are still challenges to overcome, the future of personalized medicine and lung cancer treatment is very bright. Thank you. Thank you.
Our next speaker is from the College of Agricultural and Life Sciences at the University of Wisconsin-Madison, Yara Al-Rayyan.
Hello everyone, my name is Yara Ryan. I'm currently a junior studying biochemistry in the College of Agriculture and Life Sciences, and today I'll be discussing personalized medicine through the lens of pharmacogenetics and how that can help us address healthcare disparities. Now, pharmacogenetics looks like a big word, but essentially what it comes down to is a study of how a person's genes affects their response to medications. Every person has unique genes and unique variations in their genes, and these variations can lead to people's bodies responding differently to medications. In some cases, these variations are also more common in certain racial and ethnic groups, and the goal of pharmacogenetics is essentially to try and pinpoint these genetic differences and understand them so that we can predict how people respond to their medication ahead of time. Let's take the drug warfarin, for example. Warfarin is a drug that's commonly prescribed as a blood thinner, and genetic variations in the VKORC1 and the CYP2C9 genes can significantly alter how a drug like warfarin is metabolized in the body. These genetic variations are also more common in people with Asian and African ancestry, and as a result, the dosage of warfarin or the actual drug that's used as a blood thinner might need to be altered in order to have the same effect on them. Another example of this is irinotecan, which is a chemotherapy drug that's used to treat a lot of different cancers. And genetic variations in the UGT1A1 gene can really alter how toxic irinotecan is in a person's body. This genetic variation specifically is also more common in Latino, African American, and Asian populations, and as a result, this can make irinotecan an ineffective or even a dangerous choice for the patients that have this specific variation. And knowing and utilizing pieces of information like this really underscores how important it is to personalize medicine and really optimize cancer treatments by taking into account all these different factors. Warfarin and irinotecan are just two examples of many drugs that can vary in their efficacy depending on a person's genes. And here you can see lists of other drugs that are commonly used to treat diseases that have identified gene-drug interactions by the FDA, which means that some people may need a different dosage or a different drug entirely to help treat them because of their genes. And in the future, we might identify even more genes that alter the efficacies of even more drugs. Now imagine a healthcare system that recognizes these genetic differences. Personalized medicine, driven by pharmacogenetics, can really pinpoint the genetic factors that will alter how a person's body will respond to a medication, so that we can customize and tailor treatments ahead of time. Through a blood test or even a simple cheek swab, we can really alter the treatment and personalize it to the person that in their genome. And the result is a safer and more effective treatment in a healthier world where we can really take into account all of these genetic factors and really personalize people's medicines so that they can optimize their chances of actually being effective. And this is the part that really excites me about personalized medicine, is that it can really expand the horizons of what we can do in healthcare. We learn so much and we continue to learn even more, and utilizing every piece of information and tool at our disposal is really important to make sure that every patient has the most healthy outcomes and has the tailored treatments designed just for them. This opens the door to even more research so that we can pinpoint what makes one drug right for some people and not effective for others, so that we can make the most informed and correct decision the first time around. Personalized medicine and pharmacogenetics are really the keys to a more equitable and effective healthcare system, where we can tailor treatments to what we know about a person's genome and really reduce healthcare disparities by making sure that everyone receives the care that's designed just for them. Thank you very much.
Okay, our next speaker is from Human Oncology at the University of Wisconsin-Madison, Pate Lance.
No, it is not. From current side.
Okay, let me just... can we...
Sorry about that.
That's all right. Okay. All right, there you go. You should be set.
Hi, my name is Pate Lance, and I'm a sophomore undergraduate here at UW-Madison. I'm actually in the College of Agriculture and Life Sciences. I just do some research in the Department of Human Oncology. So today I'm going to be talking to you about chromosomal instability and its implications for personalized medicine. I define personalized medicine as the creation or adaptation of existing medicines to better treat an individual or the patient's condition in a more individualized or personal manner. So, in the field of oncology and cancer treatments, as Zach had pointed out, we've been trending towards more personalized medicine for the past 2 decades or so. And while you've heard about gene sequencing, advancements in immunotherapy, or maybe even the pharmacological advancements for chemotherapies, I doubt that you've heard of the field of chromosomal instability. So, what is chromosomal instability or SIN? Chromosomal instability is the successive, chromosome missegregation events, which is when whole or parts of chromosomes don't end up where they're supposed to in mitosis. When that occurs over successive generations in mitosis, we refer to that as chromosomal instability. As you can see on my figures, the 3 images on the far left are examples of chromosomal instability. Because there's different types of chromosome missegregation events, there's different types of SIN, and it's insinuated with different levels. If you look at the 2 images on the far left, you can see examples of multipolar mitosis, evident by the fact that normal diploid cells are dividing in more than 2 ways, which isn't good. And in the middle, we have a misaligned chromosome cell. That should be in metaphase, but we can see all those chromosomes in blue there aren't in the middle like they're supposed to. So that's not typical of metaphase. Chromosomal instability is present in 60 to 80% of all tumors, so it's very prevalent in cancer. And the presence of SIN insinuates a variety of different consequences in cancer. At low levels, it's beneficial to cancer. It can increase in tumorigenesis, which is the formation of tumors. It can increase resistance to certain treatments, and it increases cell fitness. But at higher levels, not even cancer cells can handle the genetic variants, or more specifically, the loss of essential chromosomes. And we can see that in the figure on the right, which is the proposed model. Certain gene adaptations and certain different types of cell lines have different tolerances of SIN, but the bell curve shape remains the same. So as a field of researchers, we want to exploit this mechanism and increase SIN past the tolerable threshold to increase cell death. A lot of mechanisms cause SIN because there's a lot of different mitotic errors that cause it, but certain cancer treatments can also cause SIN, the main one being radiation therapy. So by better understanding what causes SIN and what treatments cause SIN, we can identify ways to increase SIN past the tolerable threshold and adjust radiation dosages or adjust dosages to create more personalized medicine. Radiation, as an example, if we sequence a tumor in a patient and find that they have high baselines of SIN, then we can potentially afford to give that patient less radiation therapy to achieve the same results, which would obviously dramatically lessen the amount of side effects that that person has to experience. We're still a number of years from putting this into clinic, but the implications of chromosomal instability are a very real and very novel aspect. So thank you for listening to my presentation.
And the last presentation in our undergrad group is from the Department of Biomedical Engineering at the University of Wisconsin-Madison, Shreya Sridhar.
Hi everyone, my name is Shreya Sridhar and I'm a senior studying biomedical engineering. I'm a part of the cardiovascular biomechanics lab, and one aspect of our research is predicting the mechanical failure of cardiovascular tissues. In the United States, someone has a heart attack every 40 seconds. A lot of people here may know someone who's suffered from a heart attack. They happen when there's damage to heart muscle, otherwise known as myocardium. Predicting rupture of myocardium is very important for developing novel treatments and better treating a patient post-rupture. To better predict this mechanical failure, we use experimental and computational data. I specifically use experimental techniques to understand the mechanical properties of these cardiovascular tissues. To understand these mechanical properties, we need to dig a little bit deeper into what actually makes up these tissues. Collagen is the most abundant protein in your body and provides mechanical and structural properties to these tissues, including cardiovascular ones. Studying how these collagen fibers move in different conditions and how they're oriented can give more insight into the mechanical properties. Collagen can be imaged in many different ways, including histology, second harmonic generation, and more. But a big aspect that these imaging techniques lack is being able to take a full field image of a tissue in real time. We use a novel method called quantitative polarized light imaging, or QPLI, that utilizes circularly polarized light that passes through the sample and highlights the collagen fiber orientation and the degree to which it's oriented in that direction. This is what an example segment of a pig heart valve looks like on the left before being imaged, and that's what it looks like on the right after being imaged. You can see the angles at which the collagen fibers are oriented and the degree to which it's oriented in that direction. This can ultimately provide very meaningful information on the material symmetry and help determine the mechanical properties of that tissue. Now, to better understand the collagen alignment in loading conditions similar to a working heart, we need to use this method of imaging while performing a more traditional mechanical test. This is an example of an equibiaxial test, which pulls the heart tissue equally in the XY plane. Our hypothesis is that as these different forces are being applied to these tissues, the collagen alignment is rotating, and that's what's causing that rupture. Overall, understanding more about the mechanical properties of these cardiovascular tissues in loading conditions similar to a working heart in the body will ultimately help in the prediction of heart failure and improved treatment for patients. Thank you.
Okay, at the end of the session, we'll announce the winners for both sections, but until then, we're going to move on to our graduate students. So this is the third year that we've done graduates and postdocs and medical students can all send in their 3-minute videos to be judged to compete in this competition. We've partnered with the College of Life Science Communications here at the University of Wisconsin-Madison to provide workshops and videos to help students do this, because it may seem easy to discuss science like this, but it's actually a pretty difficult thing to try and find out exactly how to talk about these very, very complicated topics in ways that most people can understand. It's hard to knock out all the jargon and make it understandable. So it's a big feat, and I applaud everybody that's presenting today. But without further ado, let's go on to our graduate students, the first of which is from the Department of Food and Nutrition Technology at the University of Wisconsin-Stout, Ariba Ali.
Did you know that over 8 million tons of plastic end up in our oceans each year? Did you know that we are using 400 million tons of plastic every year? And if we keep consuming at this rate, then by 2050, we will be producing 1,100 million tons of plastic waste. Yes, 1,100 million tons. My name is Ariba Ali, and I'm a graduate student of Food and Nutrition Technology at UW-Stout. Do you know 38% of all this plastic waste comes from one-time use plastic packaging of food and beverage material? So now is the time to think to create a more sustainable alternative to plastic packaging. The packaging which can be biodegradable. The packaging which can be cost-effective. The packaging which can help us extend the shelf life. And most importantly, the packaging which can help us save the environment. So our journey began with this simple idea. What if we could create something that could replace plastic? And it turned out that we can use milk proteins. Yes, you heard it right. Milk proteins called whey protein isolate and glycerol as a plasticizer, 2 components that would create biodegradable films capable of replacing plastic. And this whey protein isolate serve as an excellent carrier of such components which are incorporated in films, coatings to strengthen the barrier properties and to enhance the functional properties that are antioxidant and antibacterial. And the packaging system sourced from dairy protein serves as an outstanding shield against the physical and microbial contamination, thus playing a very important role in shelf life extension and quality maintenance of varied food products. So over the time, we formulated 4 different films of varying amounts of these compounds, whey protein isolate and glycerol. We tested the films, characterizing their physical and chemical properties, and we found out that the films made with 60% and 80% glycerol were of highest quality, while others were too brittle to be formed as a packaging material. The cost of these films would be approximately $0.5 to $2 per square meter. We really believe that our research have a great potential to have a better impact on food packaging selection and environmental safety. We hope that our work will inspire others to think creatively and find new ways to protect our environment. Thank you.
Okay, and our next speaker is from the Department of Physiology at the Medical College of Wisconsin, Melissa Elbis.
Hi, my name is Melissa Elbis, and I am a PhD student at the Medical College of Wisconsin in the laboratory of Dr. Matthew Hodges, where we have a rat model of epilepsy. Epilepsy is a common and chronic neurological condition that affects millions of people worldwide and is characterized by repeated and unprovoked seizures. But what is a seizure? Well, the brain has billions of neurons that communicate with each other by sending and receiving electrical impulses, and a seizure is a sudden surge of abnormal electrical activity in the brain. And 1/3 of patients experience repeated and uncontrolled or drug-resistant generalized tonic-clonic seizures, or GTCS, which is one type of seizure that involves the entire brain and is characterized by these stages, including the tonic stage, which is a stiffening of the muscles, the clonic stage, or the rapid jerky movements, and the post-seizure stage, which is a suppression of sensory and motor function. These patients are also at a higher risk for sudden unexpected death in epilepsy, or SUDEP, which is defined as a sudden and unexpected death in a person with epilepsy without any other apparent cause of death. And this is really important because SUDEP is a leading cause of death in this patient population and commonly affects young adults, such as these 2 celebrities who died of SUDEP. However, the actual mechanisms that lead to SUDEP are not known, and there's currently no reliable way to predict or prevent SUDEP. So this begs the question of how we can study seizures and SUDEP. Our lab has a rat model with a genetic mutation in a gene that contributes to establishing the electrical impulses that neurons use to communicate, like we talked about earlier. And when you genetically delete and inactivate this gene in a rat, they become susceptible to sound-induced seizures. And once we established this unique rat model of seizures, we began to study what happens with breathing, because in humans, seizures disrupt normal breathing processes, and this is thought to contribute to SUDEP. And this has been shown to be consistent in our model, in which repeated seizures causes progressive respiratory suppression and mortality. Also in humans, seizures disrupt normal cardiovascular function, which is what I am currently studying in this model. And our current data suggests that repeated seizures cause a cardiorespiratory suppression prior to death, similar to that in humans, and so therefore this model may have a high relevance to human SUDEP as we continue to understand the mechanisms of cardiorespiratory suppression to identify biomarkers to predict SUDEP, with the ultimate goal of identifying and testing therapeutic targets to reduce the risk of SUDEP in patients with epilepsy. Thank you.
Our next talk is from the Department of Communication Sciences and Disorders at the University of Wisconsin-Madison, Nimesha Didulani.
Hi, I'm Didulani Dantanarayan, a PhD student in the Binaural Hearing and Speech Laboratory of Dr. Litovsky at University of Wisconsin-Madison. I'm studying about children with hearing loss who are using cochlear implants. Cochlear implants provide hearing for children who are deaf. Right now, there is an emerging number of children who are using cochlear implants for both of their ears. So, there is a high number of children who are using cochlear implants for both of their ears. So because of that, it is really important to encourage children to use cochlear implants in case they have hearing loss for both of their ears, because that will help them to localize sound and also to understand speech in difficult listening situations like everyday noisy environments. So when talk about real life listening environments, we know that they are very noisy and complex. So in order to perform well in real life listening environments, just like, perfectly differentiate between the speech that we want to hear from the unwanted noise, that cannot be done in case if the hearing loss is exist. So this causes kind of a confusion. So imagine if speech of interest and noise is coming from the same direction. This leads to cause confusion that ultimately cause poor speech recognition. However, when talk about speech of interest and separated it from noise, this leads to make easier understand of speech that ultimately improves speech recognition. So when we talk about this concept with respect to children who are typical hearing, they can get the advantage of this separation of speech of interest and noise, and that will improve their speech understanding really well. So if we apply this concept to children who are using cochlear implants, we cannot see the exact benefit that we can see from children who have normal hearing. So the question is, why can't they receive the same benefit just like their typical hearing peers, even though they have been fitted with cochlear implants with the most advanced technology? So this is what I'm interested to investigate in my study, because it is really important to figure out why they can't take the advantage of using cochlear implants for both of their ears. So this inability to get this separation of speech of interest and noise can be attributed to several factors, just like neural integrity. So they have like lack of neural integrity of their auditory system. And also they do not have very well functioning auditory nerves in both of their ears. And it could be because of their cognitive skills, just like memory, attention, cognitive flexibility. And also it could be because of their amount of language skills that influence their speech recognition in noise, and also it causes difficulties that they have when separating speech of interest from noise. In addition to that, the limitations of their hearing devices that are unable to process speech signal at the same time can also cause these difficulties. So because of that, it is really important to further investigate speech sound processing in children with cochlear implants, and also to further understand how auditory maturation, cognitive skills, and language skills help to improve their communication skills. Thank you very much.
All right, our next speaker is also from the Department of Physiology at the Medical College of Wisconsin, Mark Vanden Avant.
All right, hello, I'm Mark Vanden Avant, a graduate student in the laboratory of Dr. Aaron Gertz at the Medical College of Wisconsin. And I'm here to talk to you about the role DNA looping is going to play in blood pressure regulation. So first, a little bit of background. We have our DNA. DNA encodes for RNA, and RNA is sort of like the blueprints for proteins. We are interested in how DNA can regulate RNA expression. So our genome is made up of DNA. It's made up of these non-coding and these coding regions. These non-coding regions can be known as junk DNA, and these coding regions is known as genes. And regulating how much a given gene is expressed is very important. There are several mechanisms that can influence gene regulation. So we are interested in how this junk DNA can affect gene expression. We know nearly every cell in our body has DNA and is packaged in the nucleus. So the length of DNA, if you spread it out end to end, is roughly 6 feet long. So how can we fit all of this DNA in every single cell? So the DNA needs to be coiled and then coiled and then coiled again, where it's supercoiled. And that's where these loops form. So these loops are important because it brings areas or regions that are far away to interact with maybe a specific gene of interest. So these these areas may contain promoters or enhancers that can influence gene expression. And maintaining these loops are important for proper gene expression. So there are these proteins that their main function is to maintain these loops or help form these loops. And if one of our main questions in our lab is, can disruption of these DNA loops affect gene expression and ultimately blood pressure? Currently, there are no examples that we are aware of. So our lab is studying the DNA looping in the context of high blood pressure or hypertension using specific animal models. Hypertension can lead to heart attacks and many other adverse effects. But the best thing about my project is that this can be expanded to look at or study any disease. Typically, there's some sort of genetic component for every disease, and figuring out these mechanisms can be very beneficial. And with that, I just want to say thank you, and I am Mark Van Aven from the Medical College of Wisconsin.
Okay, and last but not least, from the Department of Chemical and Biological Engineering at the University of Wisconsin-Madison, Nathan Blaylock.
Happy Tuesday, everybody. So my name is Nathaniel Blaylock, and I work in Dr. Philip Romero's lab here at Wisconsin. And today, let's explore how we can use machine learning to solve nature's protein mysteries. So let's start with the central dogma of biology. DNA can be transcribed into RNA, which can then be translated into proteins. Proteins, in turn, have a certain measurable fitness that we are often interested in. Protein fitness, specifically, is a measurement of a protein's function, and this is affected by protein properties such as activity and stability, experimental conditions such as temperature and pH, and biological constraints that affect protein expression. This complexity is what makes proteins one of nature's most intriguing mysteries, and it's also what makes them so challenging to study and engineer. Machine learning enables us to navigate this complexity much more effectively than ever before, especially compared to traditional protein engineering methods. So, just like a detective gathers clues and analyzes these clues to solve mysteries, machine learning enables us to do the same. For example, we feed experimental data and evolutionary data of functional proteins that occur in nature to the neural network. This is like giving our detective all the case files and evidence. The neural network then learns from this data and can generate a protein fitness landscape model that enables us to learn and decipher how protein properties, experimental conditions, and biological constraints affect protein fitness. In essence, machine learning acts as a detective, piecing together the clues to solve the intricate mysteries of protein fitness. So what does this look like in the real world? This is an example from my research. I fed experimental and evolutionary data for polyketase cyclase proteins, a key enzyme in the cannabinoid synthesis pathway, into neural networks. The neural networks learn from this data to produce a polyketase cyclase fitness model where optimal fitness we could learn from this spot on the model, protein properties and experimental conditions and biological constraints. And from this information, I was able to engineer a polyketase cyclase variant with high cannabinoid production. So why does that matter? Well, I introduced my engineered polyketase cyclase variant into yeast. And then in a bioreactor, we can produce therapeutic cannabinoids. These cannabinoids have potential for treating neurodegenerative diseases, gastrointestinal diseases, and many chronic illnesses, even cancer. And this is why machine learning specifically has an ability to revolutionize healthcare, because it has an ability to impact many different avenues of medicine. So in conclusion, Machine learning is really cool, and it really can help us in the future to develop protein drugs for a variety of medicines.
Okay, before we announce the winners, I just wanted to briefly thank our judges. Our judges for this competition are made up of board members of BioForward and former board members of BioForward. These people are handpicked because they really enjoy working with students and going through all of these projects and really providing good feedback. So thank you to the judges for all the work that you do. So, I'm going to announce the winners of our undergraduates first, from 5th all the way up to 1st. Remember, these are the finalists. There were several other entries that are not here today. These are all winners. And then I'll have you hold your applause until we've have all 5 standing up here, and then we'll give them our appreciation. So, in 5th place, Pate Lance. 4th place, Zach Spears. 3rd place, Shreya Sridhar. 2nd place, Mia Kroll. And 1st place, Yara Al Rayyan. What's that? Sure. You can get your seats, but don't go anywhere, because I have certificates and your cash rewards here today. So, all right, and now to the graduate students. In 5th place, we have Mark Vanden Avond. In 4th place, Nimesha Didulani. In 3rd place, Melissa Elvis. In 2nd place, Nathaniel Blaylock. And 1st place is Ariba Ali.